Welcome to this edition of Texas Fossil Hunting by William Thompson. Hope you have fun watching this video. We've got a beautiful, beautiful, tiny fossil from Texas. It's from Central Texas. The only place I know where to find it is around Canyon Lake. And this fossil is a sea urchin, a fossil echinoid called Codiopsis Pulchella. The name Pulchella came from the Latin word for beautiful little. I'd say that's pretty appropriate. This holotype and the echinoid that is photographed here that came from Ed and the one I found and the few others found were all found in Central Texas, Comal County. It was in the Lower Cretaceous Trinity group. The formation is known as the Glen Rose, and the holotype was found off Rebecca Creek Road, which is west of Canyon Lake, and the specimen that Ed found and I found were found south of Canyon Lake. This sea urchin was described and published in 2009 by Andrew Smith and William Rader. Andrew Smith was with the Natural History Museum in London. William Rader lives in Austin, Texas. The name of the publication was Echinoid Diversity, Preservation Potential, and Sequence Stratigraphical Cycles in the Glen Rose Formation, which is early Albian, early Cretaceous, Texas, USA. That is a mouthful. At the time of this paper, the specimen described was the only known specimen, and it was designated as the holotype. And the holotype is given a number at a depository, and they picked the museum in London as the depository. So that holotype specimen is in London currently, and people can go to London and with permission look at it and study it. It has a number BM8NHEE13472. Like I said, it's a small echinoid, 12.5 millimeters by 7.7 millimeters. So all the specimens that I have named, I used the University of Texas invertebrate lab as the depository. The pictures that you're looking at of this Codiopsis are considered a hypotype because I photographed it and published it in my 2016 book, Fossil Echinoids of Texas, a monograph of fossil sea urchins, which at that time was a list with photographs and descriptions of every known echinoid known in Texas as of that time. And again, that specimen was found by Ed Elliott. The echinoid that's photographed here, again found by Ed, is smaller than the holotype. It measures 7.2 millimeters in length and 4.6 millimeters in height. The one I found is even smaller. So you've got to get on your hands and knees and have some good glasses and crawl around in order to find these, because they are tiny. This is a lateral view of Ed's echinoid, not of the holotype. That is the measurements of the holotype. This echinoid, I cleaned it. You can still see there is some matrix still on it, but beautiful echinoid with no defects, a wonderful specimen. Well, let's go ahead and point out the features of this sea urchin. It's high dome shaped with a flat bottom. Very distinctive look. When you find it, you immediately know which echinoid you have. A very important feature of Codiopsis is the lack of tubercles on the surface. The tubercles on the bottom are a standard tubercle that holds a spine. What you see on the top are called glassy knobs or glassy pustules, and they're sort of randomly placed along the surface of the echinoid. These pustules are very small. In some specimens, like Codiopsis doma or Stevensoni, some of them are actually broken off, and therefore the top of the echinoid is pretty much naked. So far, all the specimens that we found in the Glen Rose are complete with the pustules. Another important feature of Codiopsis is the tubercles, which are in this specimen at the bottom, are imperforate and non-crenulate. That's two features that we're going to go over. The photo on the left is this specimen, and it's a close-up of the tubercles on the bottom, which are imperforate is one of the features, and non-crenulate is the other feature of these tubercles. On the right side, I put a Laureolea, also from the Glen Rose, and a close-up of tubercles on it. And you'll see, if you look at the blue arrow on the very top of the tubercle, you will see a perforation in the tubercle. If you look at the red arrow, you'll see these indentations or bumps around the mamelon of the tubercle. That is what's considered crenulate. This is an important feature to know when studying and determining different species and genuses of echinoids. Another feature to study on sea urchins is the apical disc. The apical disc on this specimen is small. It's in the top center. We'll talk about that further. I did a rough outline of the apical disc system, which is the reproduction part of this animal. In the center is the periproct, or anus, and it's surrounded by 5 large genital plates and 5 smaller ocular plates. This is called dicyclic because there's 2 circles. The genital plates are in the inside circle and the ocular plates are in the outside circle. On the outer edge of all 5 genital plates are a little circle which is called a gonopore, and this is part of the reproduction system. So a lot of times people say, how do you know a juvenile? If these are closed, then it's probably a juvenile. If they are open, then it's an adult. The white circles that I circled in red are glass pustules. So there is a pustule on all 10 of the plates in the apical system. All sea urchins have a 5-sided star symmetry to the sea urchin. That's made up of 5 large areas called interambucral areas made up of plates, and those plates are usually large. and then there's 5 areas of ambulacral plates, 5 areas of plates that are usually smaller. I pointed out these areas on this specimen. Also, when you look at this, the ambulacral area is a little sunken on the surface, which helps you identify this species. Another important way to identify sea urchins and this specimen of sea urchins is by counting and looking at the arrangement of pore pairs. The pore pairs are used to direct food towards the mouth. A lot of small spines are used for that. There's all types of soft parts on sea urchins which are not preserved because they were soft. So on either side of the ambulacral plates are rows of pore pairs. So they come in pairs, 2 to a pair. So there's the picture here shows the ambulacral area with pores on the left and pores on the right. On the left, I got a dotted arrow showing that those all line up to make one line of 2 sets of pores. Since it's one line in that set, it's called uniserial. On the right side, I've got a bracket showing you for every 3 sets of pore pairs, which is 6 pores, which line up with each ambular plate, and usually each ambular plate has 1 tubercle. So for each plate or each tubercle, you see 3 sets of pores, or 6 pores total. That is considered trigeminate. Another important feature in identifying your sea urchins is the mouth or peristome. This one is extremely large and takes up almost the whole bottom. This is pretty uncommon, but it's a definite feature of this sea urchin. On some species, a common comparison is the size of the apical system versus the size of the peristome. So obviously the apical disc is much, much smaller than the peristome. One of the most beautiful aspects of the Codiopsis, if you look at the bottom, the tubercles line up in such a way as to make a V shape. The hemicodiopsis means there is 1 row of tubercles for each plate in the interam, so it forms 1 V. A normal Codiopsis, such as the one on the right that is called the Codiopsis doma, there are 2 tubercles on every plate in the interam, so it forms 2 V's. There are some Codiopsis being studied currently which have 3 rows of tubercles in each interam plate, so it forms 3 V's. Really a distinct feature. Well, we made it to the end. I hope you enjoyed the presentation. I hope you learned something about fossil sea urchins as a whole and the different parts of them. And also using literature to help you find the specimens you're looking for. When this paper came out in 2009, all the local amateur paleontologists including myself, started tearing through the document and looking for these specimens. Had it not been for this paper, we may not have found it ourselves. But several collectors do have this specimen, and it's a beautiful sea urchin to add to any fossil collection. So happy hunting. Hope you enjoyed the presentation.